Lecture Notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. 5. The mitotic cell cycle. When the body cells reached a certain size, they divided into two cells. Nuclear division occurs first, called as karyokinesis, followed by division of the cytoplasm called cytokinesis. This mitotic cell cycle of eukaryotes involves DNA replication followed by nuclear division. This ensures genetic uniformity of all the daughter cells. 5.1 Replication and division of nuclei and cells. Cells can be made only when existing cells divide. There are two types of cell division in eukaryotic cells, mitosis and meiosis. Cell division in prokaryotic cells, such as bacteria, does not involve mitosis or meiosis. Dividing onion cells can be viewed by preparing a temporary mount of squashed root tip, stained using acetoorcein. Rapidly dividing cells undergo a cell cycle. The G1, S and G2 phase of the cycle represent interphase. The M phase of the cycle represents mitosis. DNA replication occurs in the G1 phase of the cell cycle. During nuclear division, chromosomes become visible and are seen to consist of two chromatids held together by a centromere. Each chromatid contains one DNA molecule. During cell division, the nucleus divides first, followed by division of the whole cell. Division of a nucleus to produce two genetically identical nuclei is achieved by the process of mitosis. Mitosis is divided into four phases: prophase, metaphase, anaphase, and telophase. The period from one cell division to the next is called the cycle. It has four phases. G1 is the first growth phase after cell division. S phase is when the DNA replicates. G2 is a second growth phase. M phase is when nuclear division takes place, followed by cell division. 5.1-1 Structure of a chromosome limited to DNA, histone protein, sister. Chromatids, centromere and telomeres. Lecture Notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. Chromosomes are made of chromatin. Chromatin consists mainly of DNA wrapped around basic protein molecules called histones. Chromosomes are the structures having an X-like shape, and they are packed in a condensed from inside the nucleus of the cell. The genetic information of the cell is contained inside the chromosome. The chromosomes are responsible for transferring genetic information from parent to the offspring. 5.1-2 Importance of mitosis in the production of genetically identical daughter cells during growth, replacement, repair, and asexual reproduction. Mitosis produces cells that are genetically identical to each other and to the parent cell. Mitosis is used in growth, repair, asexual reproduction, and cloning of cells during an immune response. Cancers are tumors resulting from repeated and uncontrolled mitosis. They are thought to start as the result of mutation. 5.1-3 Mitotic cell cycle, including interphase, growth in G1, G2, and DNA replication in S phase, mitosis and cytokinesis. Mitosis consists of several stages: prophase, metaphase, anaphase, and telophase. Interphase is the period between mitotic divisions. In prophase, chromosomes condense to become shorter and fatter. We can see them using an optical microscope. Each chromosome has already been copied. The two. Lecture Notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. Copies, called sister chromatids, are held together by a centromere. The nucleus disappears and a spindle of protein fibers forms during prophase. In anaphase, each centromere divides, separating sister chromatids. Contraction of fibers in the spindle pulls the two sister chromatids of each pair to opposite poles of the spindle. In telophase, the separated chromatids, now called chromosomes again, uncoil to become long and thin again. Division of the cytoplasm, cytokinesis, usually occurs. In cells without a cell wall, this is done by pinching the cytoplasm in the middle of the cell. In cells with a cell wall, new cell wall is secreted and separates the two new cells. Interphase is the time between cell divisions having G1, S and G2 stages. 5.1 to 4 Role of telomeres in preventing the loss of genes from the ends of chromosomes during DNA replication. As the cells of our body divide and renew themselves, telomeres protect the ends of chromosomes and allow our genetic material to be copied correctly. But every time a cell divides, telomeres lose base pairs of their DNA. So as we age, telomeres tend to get shorter and shorter. When telomeres are completely worn away, chromosomes can break, fuse with other chromosomes, or mutate. This leads to cell dysfunction, disease, and cell death. 5.1 to 5 Role of stem cell in cell replacement and tissue repair by mitosis. Many specialized cells lose the ability to divide, but certain cells known as stem cells retain this ability. Stem cells are essential for growth from zygote to adult and for cell replacement and tissue repair in the adult. 5.1 to 6 Uncontrolled cell division resulting in the formation of a tumor. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. Cancers are tumors resulting from repeated and uncontrolled mitosis. They are thought to start as the result of mutation. Growth of a multicellular organism is a result of cells dividing to produce genetically identical daughter cells. Cancers demonstrate how important it is that cell division is precisely controlled. As cancers arise due to uncontrolled mitosis, cancerous cells divide repeatedly and uncontrollably, forming a tumor, an irregular mass of cells. Cancers start when changes occur in the genes that control cell division. A change in any gene is known as a mutation. If the mutated gene is one that causes cancer, it is referred to as an oncogene. Mutations are common events and don't lead to cancer most of the time. Most mutations either result in early cell death or result in the cell being destroyed by the body's immune system. As most cells can be easily replaced, these events usually have no harmful effect on the body. The mutations that result in the generation of cancerous cells do not result in early cell death or in the cell being destroyed by the body's immune system. This means that the harmful mutation occurring in the original cell can be passed on to all that cell's descendants. A typical tumor contains around 1,000 million cancerous cells by the time it is detected. Carcinogens are any agents that may cause cancer, e.g. UV light, tar and tobacco smoke and X-rays. If the agent causes cancer, it is described as carcinogenic. Some tumors, such as warts, do not spread from their original site. These are known as benign tumors and do not cause cancer. Some tumors spread through the body, invading and destroying other tissues. These are known as malignant tumors and cause cancer. Malignant tumors interfere with the normal functioning of the organ/tissue in which they have started to grow. For example, they may block the intestines, lungs, or blood vessels. Malignant tumor cells can break off the tumor and travel through the blood and/or lymphatic system to form secondary growths in other parts of the body. The spreading of cancers in this way is known as metastasis. Metastasis is very dangerous as it can be very difficult to detect, locate, and remove secondary cancers. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. Diagrammatic illustration of tumor formation. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. 5.2 Chromosome behavior in mitosis. The behavior of chromosomes during mitosis can be observed in stained preparations of onion root tips. Choose a very long root and cut off about a 0.5 centimeter, a quarter inch, of the root tip. Place the tip on a slide and add 2 drops of 0.5 m hydrochloric acid, HCl, to the tip. Let the root tip soak in the HCl for 10 minutes. After 10 minutes, use a paper towel to carefully soak up all of the HCl. 5.2 to 1 Behavior of chromosomes in plant and animal cells during mitotic cell cycle, including different phases. Events that occur during the stages of mitosis. Stage of mitosis. Main events that occur. Prophase. Chromosomes start to coil, becoming shorter and fatter. Nuclear envelope disappears. A network of protein fibers, the spindle, forms in the cell. Metaphase. Chromosomes line up on the equator of the spindle. Fibers of the spindle attach to a region of each chromosome called the centromere. Anaphase. Spindle fibers contract and pull the 2 copies of each chromosome to opposite ends of the spindle. Telophase. The 2 sets of chromosomes form new nuclei. During this process, the chromosomes become long and thin again, and new nuclear envelopes form around the nuclei. The cytoplasm usually divides to form 2 new cells. Interphase. The daughter cells grow more cytoplasm and get on with their normal activities. The cells make copies of their chromosomes, by DNA replication, ready for their next division. Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 03004610457. Prophase. Chromatin condenses to, duplicated, chromosomes. Nucleus disappears, nuclear envelope fragments, centriole pairs migrate toward opposite poles, polar microtubules are assembling. Metaphase. Nuclear envelope fragmentation is complete, spindle formation is completed, duplicate chromosomes are aligned at equator, kinetochore microtubules of sister chromatids point to opposite poles. Anaphase. Quicker phase of mitosis. The centromere divided into 2. Spindle fiber pulls the daughter chromatids towards the opposite poles of the cell. Telophase. Chromosomes uncoil to chromatin. Nuclear envelopes reform. Nucleoli reappear. Daughter nuclei are diploid. Cytokinesis is nearly complete. Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 03004610457. Interphase. The daughter cells grow more cytoplasm and get on with their normal activities. The cells make copies of their chromosomes, by DNA replication, ready for their next division. 5.2 to 2 Photomicrographs, diagrams and microscope slides of cells in different stages of mitotic cell cycle. Steps in the production of the root tip squash. This process enables us to see cells dividing by mitosis. Steps in procedure explanation. Use a scalpel to cut about 3 mm from the tip of a growing root. Put the cut root tip onto a watch glass. Dividing cells are found only at the very end of the growing root. Behind the root tip, cells have elongated and differentiated into mature, non-dividing cells. Wearing gloves, add 1 mol DM3-acetic acid, also known as ethanoic acid, to the root tip. Put the watch glass onto a hot plate and warm for about 5 minutes, do not let the acid boil. The warm acid helps to soften the tissues so that cells can be separated more easily. Use forceps to put the root tip onto a glass slide. We always mount specimens on a glass slide before viewing them with an optical microscope. Add a few drops of acetoarsine stain to the root tip on the slide. The stain is taken up by chromosomes, making them easier to see. Gently break up the root tip using a mounted needle. You should attempt to spread the root tip out thinly, rather than stir the cells from the root. This separates cells. Put a cover slip over the root and cover the cover slip with a piece of filter paper. Press down on the cover slip gently with your thumb. If you press too hard, the cover slip will break. The aim here is to produce a layer that is only one cell thick. This makes individual cell easier to see. Use an optical microscope to examine the slide for cells dividing by mitosis. You will now see onion cells that have their chromosomes stained a reddish-purple color. Lecture Notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 03004610-457. Interpretation of photomicrographs and microscopic slides of cells in different styles. Identify different phases of mitosis in the above photomicrograph. Lecture Notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 03004610-457. Keywords: anaphase, asexual reproduction, bivalent, centromere, chromatid, chromosome, cytokinesis, deoxyribonucleic acid, DNA, differentiation, G1, gap1, phase, G2, gap2, phase, homologous chromosomes, interphase, metaphase, mitosis, mitotic spindle, prophase, sister chromatids, S, synthesis, phase, stem cell, telomere, telophase, tumor. Chapter Glossary. Anaphase is the third stage in mitosis where the chromosomes are pulled apart to the poles of the cell by the spindle fibers. Asexual reproduction is the production of genetically identical offspring from one parent through the process of mitosis. Bivalent is a pair of homologous chromosomes. Centromere is a structure on a chromosome that holds the sister chromatids together. Chromatid is one strand of a replicated chromosome. Chromosome is a structure consisting of a long, coiled molecule of DNA and its associated proteins, by which genetic information is passed from generation to generation. Cytokinesis is the division of the cytoplasm at the end of mitosis to produce two new daughter cells. Deoxyribonucleic acid, DNA, is an information-storing molecule made up of deoxyribonucleotide monomers joined by phosphodiester bonds to form a double helix. Differentiation is the process where a cell develops certain features so that it is specialized to carry out a certain function. G1, gap1, phase is the first growth phase in interphase during which the cell synthesizes proteins and RNA, duplicates its organelles and increases in size before DNA replication in S phase. G2, gap2, phase is the second growth phase of interphase during which the cell continues to increase in size and synthesize biomolecules. Histone proteins are proteins that, together with DNA, form chromosomes in the nuclei of eukaryotic cells. Homologous chromosomes are two chromosomes with the same gene loci but different alleles, one inherited from each parent. Interphase is the longest stage of the eukaryotic cell cycle. It consists of G1 phase, S phase and G2 phase and occurs before mitosis. Metaphase is the second stage of mitosis in which replicated chromosomes align at the equator of the cell. The division of a cell to produce two genetically identical daughter cells. Mitotic spindle is a structure mainly consisting of microtubules that is formed by the cytoskeleton between centrioles. Chromosomes attach to the spindle via the kinetochore at their centrosome. Prophase is the first stage in mitosis where the nuclear envelope breaks down, the centrosomes move to opposite poles of the cell, the mitotic spindle begins to form and the chromosomes condense. Sister chromatids are a pair of identical chromatids formed by DNA replication, joined by a centromere. S, synthesis, phase is the second phase in the cell cycle where the DNA in the cell is replicated. Lecture Notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 03004610457. Stem cell is a type of undifferentiated cell which has the ability to divide many times and differentiate into many different cell types. Telomere is a sequence of repeating nucleotides at each end of a chromosome. During DNA replication, the end of the lagging strand either is not replicated or remains as an RNA-DNA complex. Without telomeres, the chromosome would get shorter with continued replication and some genetic information may be lost. Telomere is the final stage of mitosis in which new nuclear envelopes begin to form around the separated sets of chromosomes. Tumor is an abnormal growth of tissue resulting from uncontrolled mitosis. Short MCQs with right answers. 1. The cell cycle includes mitosis. What are features of this type of nuclear division? Forms genetically identical nuclei. 2. The diagram shows part of the organization of a section of a DNA molecule and the associated histones, P and R, in prophase of mitosis. 3. Which statement about the features labeled P, Q, and R during prophase of mitosis is correct? The linked groups of histones P and R and the associated DNA, Q, form strands that fold and twist together to form a chromatid. 4. How many copies of each DNA molecule will be found in a cell at the start of the stages of the mitotic cell cycle shown? 5. Which row describes some properties of stem cells? 6. The mitotic index is a measure of the proportion of cells that are undergoing mitosis in an area of tissue. It is calculated using the formula shown. Mitotic index equals number of cells undergoing mitosis divided by total number of cells times 107. A scientist calculated the mitotic index of areas of onion root at different distances from the tip of the root. The results are shown. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 03004610457. 8. Which statement is correct? Most of the cells undergoing cell division are closer to the tip of the root. 9. Which events are part of the mitotic cell cycle? 1. Interphase. 2. Telophase. 3. Cytokinesis. 10. The estimated total number of red blood cells in the human body is 2.5 times 1013. It is estimated that, each day, 2.5 by 1011 red blood cells are removed from the circulation and are replaced by stem cells in the bone marrow. Which percentage of the total number of red blood cells is replaced each day? 1%. 11. Human chromosomes have different parts. Some parts are more numerous than others. Which parts are listed in order from most numerous to least numerous in a human blood cell? Histone protein telomere DNA molecule 12. DNA molecules in a eukaryotic cell must be packed compactly because their combined length is much greater than the diameter of the nucleus, which structures are required. During the process of packaging DNA molecules compactly, histone proteins. 13. 3 parts of a chromosome and their functions are listed. Which part is matched with its correct function? Part function histone proteins holds 2 chromatids together. 14. Which description of chromosome structure is correct? The very long DNA molecule of each chromatid is coiled and held together by proteins called histones. 15. Some parts of a typical human chromosome are more numerous than other. Which of the parts are listed in order from the most numerous to the least numerous? Histone, telomere, DNA molecule 16. Which is not a description of a gene? Any section of a molecule that has 2 strands, each with a sequence of nucleotides that are complementary to each other and are held together by hydrogen bonding 17. What will be present in each chromosome at the end of the G2 phase of the cell cycle? 2 molecules of DNA 18. The jellyfish, Turritopsis domii, is described as being immortal. If T. domii is not eaten by predators or diseased, it seems to be able to live forever. There is no way to determine the biological age of a T-domii individual. Which feature of the cells in T-domii could explain these observations? An ability to restore telomeres to their original length. 19. During the cell cycle, the mass of DNA in a cell doubled. At which stage in the cell cycle is the mass of DNA in a cell reduced? Cytokinesis. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. 20. Which metabolic processes will be very active in a cell that has just completed cytokinesis? ATP formation, protein synthesis. 21. Which of these events is part of mitosis? Telophase. 22. During which phase of the cell cycle does DNA replication take place? S23. What is the correct sequence of stages in the mitotic cell cycle? G1S G2 mitosis cytokinesis. 24. The protein P53 is produced in a cell in response to DNA damage. This protein stops the cell cycle for a short time just before the DNA is replicated, so that the DNA can be repaired. At which phase of the cell cycle will this stop occur? G125. Which statements about the cell cycle are correct? 1. The cell cycle consists of both interphase and mitosis. 2. DNA replication takes place in interphase. 3. A cell can remain in interphase for several months. 26. Which is always true of cytokinesis. Cell structures are divided between two cells. 27. Telomeres prevent the loss of genes from the ends of chromosomes during DNA replication, but they become shorter each time they are copied. In cancer cells or stem cells, the telomeres remain the same length. Which statement is correct for all human cells? If telomeres become too short, a cell may stop dividing. 28. Which description is correct? A telomere is a sequence of DNA nucleotides, such as GGGTAA, repeated many times and found at the ends of each chromatid. 29. How many of the listed structures typically contain genetic material that has telomeres? Nucleus. 30. During telophase of mitosis, a scientist stains the chromosomes of a diploid animal cell with a fluorescent dye to observe the telomeres. This cell has 22 chromosomes. How many telomeres will the scientist observe in one of the nuclei? 4431. During prophase, a scientist stains the chromosomes of a diploid animal cell with a fluorescent dye to stain telomeres. This cell has 32 chromosomes. How many telomeres will the scientist observe? 128. 32. Which cells contain telomeres that are longer than those in a helper T lymphocyte? Secreting cytokines. 1. Bone marrow stem cells. 2. Activated memory B lymphocytes. 33. In which stage of the cell cycle are telomeres needed to prevent the loss of genes? S phase. 34. What happens to the telomeres during mitosis of a cancer cell? They stay the same length. 35. The enzyme telomerase prevents loss of telomeres after many mitotic cell cycles. Which cells transcribe a high concentration of telomerase? Activated memory T lymphocytes. 36. The enzyme telomerase prevents loss of telomeres after many mitotic cell cycles. Which cells need to transcribe telomerase enzyme? 1. Cancer cells. 2. Stem cells. 3. Activated memory B lymphocytes. 37. Which statement is correct about the number of telomeres present in prophase of a human body cell? 184, as there is one telomere at each end of 92 chromatids. 38. What describes a telomere? A region of repetitive nucleotide sequence at the end of a chromatid. 39. The enzyme telomerase repairs telomeres. It stops the telomeres from getting shorter each time a chromosome is replicated. Telomerase is not normally active in human body cells, but in some diseases telomerase can be activated. In which disease is the enzyme telomerase activated? Cancer. 40. Which feature of stem cells enables them to replace cells in tissues such as the skin? They divide by mitosis to supply some cells that can differentiate. 41. A gene code for the production of a protein, p53, that binds to damaged DNA during interphase and prevents its replication. A carcinogen in cigarette smoke mutates this. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. Gene. Which statement explains why this mutation may cause cancer? Cells with no p53 allow their damaged DNA to replicate. 42. Which changes in a group of mammalian cells, dividing by mitosis, would be necessary for the formation of a tumor? 1. Mitosis is no longer inhibited by cell-to-cell contact. 2. Cells become able to divide indefinitely. 43. A mutation occurs in a gene which prevents the production of telomere 4 ASE. What is the effect of this mutation if it occurs in bone marrow stem cells? Bone marrow stem cells eventually no longer divide. 44. Uncontrolled cell division can form tumors. Which statement is correct for tumor cells only? Interphase takes less time. 45. Cancer cells may divide by far more divisions than other cells found in humans. Which statement about cancer cells is correct? They are able to synthesize the enzyme telomerase. 46. Some stem cells divide and give rise to phagocytes. Where in the human body do these stem cells divide? Bone marrow. 47. Which statements about cancer are correct? 1. Some viruses can cause cancer. 2. The mutation of genes controlling the cell cycle may cause cancer. 48. Exposure to which of the following increases the risk of developing a cancerous growth? Ultraviolet light increases risk, viruses increase risk, carbon monoxide does not increase risk, X-rays increase risk. 49. Uncontrolled cell division of cancer cells forms tumors. Which statement describes the difference between a cancer cell and a normal cell? Cancer cells have a shorter interphase. 50. What occurs during prophase in animal cells? 1. Fragmentation of the nuclear envelope. 2. Nucleolus disappears. 3. Stained chromosomes become visible. 51. What is a role of mitosis? Growth of organisms. 52. During metaphase, a scientist stains the chromosomes of a diploid animal cell with fluorescent dye to allow the telomeres to be observed. This cell has 26 chromosomes. How many telomeres will the scientist observe? 104. 53. Which features of an organism are affected by a drug that kills cells that are dividing? Mitotically. 1. Cell replacement. 2. Number of stem cell. 3. Tissue repair. 4. Tumor formation. 54. Which events listed are part of the cell cycle? 1. Interphase. 2. Metaphase. 3. Cytokinesis. 55. Some chemicals, used to stop tumor growth, work by preventing the DNA double helix from uncoiling and separating. During which stage of the cell cycle would they act? Interphase. 56. At which stages of mitosis are chromosomes composed of two chromatids that are held together by a centromere? Metaphase and prophase. 57. Which statements about mitosis are correct? 1. Mitosis results in genetically identical cells. 2. Mitosis is involved in sexual reproduction. 58. Some events in cell cycle are listed. Which events are part of mitosis? Metaphase. 59. Which features of mitosis are important for a single CE3-led organism? 1. Sexual reproduction. 2. Production of genetically identical cells. 60. Which stage of mitosis is correctly described? In telophase, chromosomes uncoil to form chromatin. 61. Which correctly describes parts of chromosome structure present during mitosis? Centromere, chromatid, telomere, region of non-coding DNA, double-stranded DNA molecule with histone proteins. Region of DNA with many short repeated sequences of bases. Lecture notes for AS Level Biology by Mohammad Rashid Chatta. Cell number 030046100457. 62. Which processes involve mitosis? 1. Forming cells at the tip of a growing root. 2. Replacing skin cells damaged by an injury. 63. Which row shows part of the correct sequence of mitosis? Chromosomes line up at the equator. Spindle fibers shorten nuclear envelope, reappears, chromosomes uncoil. 64. Which row shows the correct number of each component of a single chromosome during prophase of mitosis? Centromeres. 1. Chromatids. 2. Polynucleotide strands. 4. Telomeres. 465. The cell cycle includes mitosis. Which are features of nuclear divisions? Forms genetically identical nuclei. 66. At which stage of mitosis do these events occur? Centromeres, separate chromosomes become shorter and thicker. Anaphase, prophase. 67. Which events listed are part of mitosis? Prophase only. 68. How many copies of DNA molecule will be found in a cell at each stage of the mitotic cell cycle? G2 of interphase, 2, metaphase, 2, cytokinesis, 269. Which row shows some of the events during a mitotic cycle in the correct time sequence? Time. Nuclear envelope breaks into small vesicles. Spindle microtubules produced. Centromere of each chromosome splits. Nucleoli become visible. 70. Which of these statements about cytokinesis is always true? Cell structures are shared between two cells. 71. A student observed the cells in the growing region, meristem, of an onion root and obtained the data shown. Stage number of cells: interphase 886, prophase 73, metaphase 16, anaphase 14, telophase 11. Which percentage of cells contains chromosomes that appear as two chromatids? 8.972. How many copies of each different DNA molecule will be found in a cell at the start of each of these stages of mitotic cell cycle? G2 of interphase, 2, prophase, 2, cytokinesis, 273. Which of these events are parts of mitosis? Anaphase only. 74. Colchicine is a chemical that stops chromatids from separating during mitosis. A cell is treated with colchicine. Which stage of mitosis will this cell reach but not complete? Metaphase 75. Which statement about the behavior of chromosomes during mitosis is correct? They reach the poles of the cell and become longer and thinner. Lecture notes for AS Level Biology by Mohammad Rashid Chatta. Cell number 030046100457. 76. Chromosome telomeres are essential for DNA replication and are not completely replaced during mitosis. Substance X completely replaces telomeres during mitosis. What will be the effect of growing cells with and without substance X? With substance X, cells divide continually. Without substance X, cell division eventually slows and stops. 77. Which group of cell can all divide by mitosis? Cancer cells, lymphocytes and stem cells. 78. Which row correctly shows the behavior of the nuclear envelope, the centrioles, and the spindle during the stage of mitosis? At telophase, nuclear envelope reforms, centrioles one besides each nucleus and spindle microtubules break down. 79. Which event is part of mitotic cell cycle? Anaphase 80. Which row is correct for a human cell just before the cell enters prophase of mitosis? Number of chromatids, 92, number of molecule of DNA in nucleus, 92, spindle present, no, nuclear envelope present, yes. 81. Which processes occur in bone marrow cell that are in a mitotic cell cycle? 1. Phosphate groups bind to ADP molecules to form ATP. 2. Bonds form between nucleotides in a DNA strand. 3. tRNA anticodons hydrogen bonds with codons on mRNA 82. Which process occurs during prophase of the mitotic cell cycle in an animal cell? Separation of centrioles. 83. Mitosis is an important process for organisms. Which features of mitosis are important for single-celled organism? Asexual reproduction and production of genetically identical cells. 84. A group of chemicals used to treat cancer prevents the formation of the spindle during mitosis. Which phase of mitosis is the first to be affected? Prophase. 85. Which processes occur during prophase of the mitotic cell cycle in an animal cell? Centrioles replicate, no, chromosomes condense, yes, spindle fibers form, yes. 86. A scientist counted 22 chromosomes in each of the root cell of a xerophytic plant. What is the diploid and haploid number of chromosomes for this species? Diploid number 22, haploid number 11. 87. Which statements about a diploid cell are correct? 1. Has 2 complete sets of chromosomes. 2. Can undergo a reduction division to form haploid cells. 3. Can undergo mitotic division to allow growth to occur. 88. During which process does only mitosis occur? The production of cancerous tissue in alveoli. 89. Which is the correct statement concerning cell and nuclear division? Haploid eukaryotes can reproduce by mitosis, whereas diploid eukaryotes can reproduce by mitosis or meiosis. 90. Which processes occur by mitosis? Cloning of plasma cells and replacing damaged cells. 91. Which features of mitosis ensure that the genetic constitution of the cell is maintained? 1. The longitudinal division of the centromeres. 2. The DNA of the parent cells replicate before mitosis begins. 3. The pulling apart of the chromatids towards the opposite poles. 92. Nocortisol is a chemical used in the study of mitosis. It causes all mitotic cells to stop dividing at metaphase. Which statements correctly identify how this chemical might work? Stop sister chromatids migrating to opposite poles. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. 93. Some chemicals, used to stop tumor growth, work by preventing the DNA double helix from uncoiling and separating. During which stage of the cell cycle would they act? Interphase. 94. Immediately after which stage in mitosis in an animal cell does the cytoplasm start to divide? Telophase. 95. Which statement is incorrect for mitotic cell division? DNA is replicated semi-conservatively during mitosis. 96. What occurs during metaphase of mitosis? Chromosomes attach to the equator of the spindle. 97. What occurs in mitosis? Homologs chromosomes not make pairs and chromosomes number remains the same. 98. Which statement describes events during interphase of mitosis? Chromosomes are replicated ready for the next division. 99. For organisms undergoing sexual reproduction, a reduction division occurs before fertilization. Which reason explains why this is necessary? Prevent doubling of the chromosome number. 100. What occurs during prophase of mitosis? Chromosomes become shorter and thicker. 101. Meiosis and mitosis are two types of cell division. A cell has 20 chromosomes before it divides. How many chromosomes will it have after dividing by meiosis or mitosis? Meiosis, 10, mitosis, 2102. Which statements about diploid cells are correct? 1. Possesses 2 sets of chromosomes. 2. Undergoes a reduction division to form haploid cells. 3. Undergoes mitotic division to allow growth to occur. 103. Cell cycle includes mitosis, which are features of nuclear division. Forms genetically identical cells. 104. What describes the behavior of the nuclear envelope and the cell membrane during mitosis? Nuclear envelope breaks down, cell membrane remains intact. 105. During which stage of the mitotic cell cycle is DNA replicated? Interphase. 106. Which statement describes a cell that is capable of reproduction and belonging to a haploid organism? It will undergo cell division by mitosis during asexual reproduction. 107. Cancer cell divide out of control, forming tumors. Which statement describes the difference between a cancer cell and a normal cell? Cancer cells have a shorter interphase. 108. Which of the following is true of the cancer? Mitosis is uncontrolled. 109. Which processes involve mitosis? Growth, repair, and asexual reproduction. 110. Which structure organizes spindle formation during mitosis in animal cells? Centriole. 111. Mammalian skin cells in tissue culture were supplied with a source of radioactive thymine. At which stage in the cell cycle will the thymine be used in the nuclei? Interphase. 112. A diploid nucleus in a species of fruit fly has 8 chromosomes. How many DNA molecules are present in the nucleus at the end of interphase? 16. 113. Which cell activity must occur before prophase of mitosis can begin? Replication of DNA. 114. What happens to chromosomes in prophase of mitosis? They shorten and become visible. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 0300461045. 115. The following events take place in the mitotic cell cycle orderwise. 1. Chromosomes condense, centrioles separate and move to opposite poles of the cell. 2. Nuclear envelope disappears, centromeres arranged at equator of spindle. 3. Sister chromatids pull toward centrioles. 4. Chromosomes unwind and cytokinesis occurs.